In this first part of our lecture on the vestibular system, we're going to focus on the overall function and the different structural components that are responsible for how the vestibular system works. And so just to provide an overview, the vestibular system functions mainly as a sensor for monitoring the position and movement of our head and of course the body. Overall, it provides a sense of balance and equilibrium to seated and upright standing posture. And so when we think about equilibrium, that refers to a state of mechanical stability of the body so that we don't result in tipping either when we are standing or we're in a seated position, both statically and dynamically. And the way this works is it operates in a similar manner as the rest of the sensory system, in that specialized sensory cells will generate action potentials in a way so that it can inform the central nervous system regarding its function to monitor the position of the head, and as well as the movements of the head. These signals will eventually get conveyed to the somatosensory cortex so that we generate a conscious awareness of head position and body position as well as movement. But it also projects to various nuclei that we'll discuss shortly. And so the function of the vestibular system is to not just sense position and movement of the head, but to provide an appropriate response to perturbations in head or body position and movement. And this occurs through the recruitment of skeletal muscles. So very fast, relatively quick pathways exist between these sensors and the motor axons that will activate our alpha motor neurons to recruit skeletal muscles to produce force to reestablish equilibrium. So when there are disruptions to the vestibular system, it results in things such as motion sickness, uncontrollable eye movements. We'll talk about the reflex that exists between the vestibular system and the ocular system, as well as it will produce a sense of disequilibrium. And so our perception of our head position, movement, and body movement will be different than what we're actually experiencing mechanically if there is a disruption in the structures of the vestibular system. And so when we take a look at the vestibular system, it does form it's located in a same area as the structures that transmit auditory information. And so we see the auditory canal will transmit sound waves through the air to the middle ear, which is involved in transducing that sound wave into an electrical signal. But our concern here is with the inner ear, and we see the inner ear contains, for the most part, the structures that form the vestibular system that will eventually transmit information through the vestibular nerve portion of the auditory vestibular nerve. And the cochlea located within the inner ear is involved in the transduction of sound waves. But as I mentioned, we'll focus our discussion on that portion of the inner ear involved with balance. So there are two major components in the inner ear. One that I just touched on, the cochlea, which transduces those air waves into action potentials, and then the vestibular labyrinth, which is an elaborate arrangement of structures that are sensitive to head position and equilibrium. Common to both of these major components is the functional unit referred to as the hair cell that we'll see shortly. If we focus here on the vestibular system, the major components are comprised of the semicircular canals, the otolith organs, Scarpa's ganglion, and the vestibular nerve. And so we see here three semicircular canals that are arranged approximately at 90 degrees to each other, and these structures are involved in generating action potentials, as do the otolith organs located at the base here of the semicircular canals. And these organs, which are made up of the utricle and saccule, are also involved in generating action potentials. These action potentials get transmitted through Scarpa's ganglion, and eventually these axons and these cell bodies, these neurons, will merge into transmitting action potentials. A study published in 1992 by Sato modeled and examined the structure of Scarpa's ganglion within the inner ear. And Scarpa's ganglion can just simply be described as the enlarged portion of the vestibular nerve, which contains the cell bodies of those vestibular neurons in a similar capacity or in a similar manner as the dorsal root ganglion houses the cell bodies for sensory nerves in the peripheral nervous system. This ganglion is found deep to the temporal bone. It's made up of both superior and inferior portions that are connected by a central isthmus. And the isthmus is essentially a narrowed portion of this cluster of nerve cell bodies. They are topographically organized, which is to say that information that is transmitted from the semicircular canals will tend to travel along similar cable lines, if you will, as do those axons that get transmitted by the separate otolith organs. Because of the enlarged and superior and inferior portions of the ganglion, it's described as having a distorted hourglass shape, and it is quite small, but some rough dimensions here in terms of the volume and the height and width are described nicely in this research study. And there seems to be a pretty high negative correlation between the height and the width of Scarpa's ganglion. So the larger that height is, the more narrow the measurements for this cluster of neurons is. So the size and shape of the ganglion tends to be consistent through the lifespan, and studies do find a relative decrease in the size and the density of hair cells with advancing age. And so the consequence of this decrease in size of the ganglion, as well as a decrease in the density of hair cells contained within the sensory organs, we'll see a decrease in the speed and the magnitude of the action potentials conveyed to the central nervous system when there is a disturbance in body equilibrium or postural balance. And so that will lead to a delay, if you will, or a reduced ability to respond to equilibrium perturbations and challenges. And so the risk of falling as a function of age increases not just because of a loss of muscle strength, but because of this loss in the ability to detect and transmit perturbations to our mechanical stability. In terms of the sensors of the vestibular system, we see that the otolith organs, their function is mainly to detect gravity and head tilts, as we'll discuss shortly. These organs are particularly responsible for detecting linear motions, as opposed to the semicircular canals, which are largely involved in detecting angular motions. But as mentioned earlier, they're composed of three arching structures which are orthogonal to each other, which is to say that they are positioned at approximately right angles to each other. And that's a handy thing for us, because that allows us to be sensitive to head rotations in space, to be sensitive to rotations around three axes of movement. The vestibular nerve is a branch of cranial nerve 8. It's composed of approximately 20,000 vestibular neuron axons that arise from the semicircular canals as well as the two otolith organs. As mentioned, their cell bodies are located in Scarpa's ganglion, and their job is to transmit sensory signals that have been mechanically detected by the hair cells within the semicircular canals as well as the otolith organs. So overall, when you think of these two major components of the vestibular system, their function is to transduce mechanical energy that arises from head and body motion to electrical energy, which in the form of action potentials, which is the means by which our body communicates from one part to another. So if we start with the otolith organs, their function is to detect a change in head angle through its sensitivity to gravity. But primarily its job is to sense changes in linear accelerations. And so we ask, why acceleration? And this ties into Newton's second law of motion, which is force equals mass times acceleration. So when our body and our head experiences an acceleration, that will result in the imposition of force on these structures, and it's going to require that application of force through body acceleration to cause a change in the position of the hair cells, which is what we'll see on the next slide. The inner part of the walls of the semicircular canals as well as the otolith organs are made up of a macula, which is a sensory epithelium, and this is the structure that contains the hair cells, and the hair cells can be described as the functional unit of the vestibular system. This is where mechanical energy is transduced into electrical energy through a deflection in those hair cells. So when those hair cells become deflected, they will either discharge the associated neuron axon, or they will hyperpolarize that axon, as we'll see shortly. Specific to the otolith organs, the saccule, the sensory epithelia are generally oriented in a vertical manner, while the utricle epithelium is generally organized in a horizontal position, and that allows us, as we discussed with the semicircular canals, to be sensitive to linear accelerations both in a horizontal and vertical manner. Key to the sensory epithelia are what are referred to as ear stones, otoconia, which are these small calcium carbonate crystals that range anywhere from 1 to 5 microns in diameter. And in this illustration, you see that the otoconia sit on top of a gelatinous cap, and contained within this gel-like structure are the projections from these hair cells. And the ear stones are the link really between the detection of head movements and transmission through the gel to the hair cells. And these forces have to be transmitted to the kinocilium, which are basically a single enlarged end of these hair cells. And so this force, as I mentioned, comes from a change in head angle or accelerations. When we're talking about the otolith organs, we're talking about linear accelerations and not spinning types of movements. So the otoconia, the crystals that sit on top of the cap, which then will transmit, when the otoconia are disturbed, that will transmit through as a force through the gel that will cause a deflection of these hair cells, and when these hair cells are deflected, it will open up ion channels that will eventually discharge through vestibular nerve axons. And one last point to make here is that the hair cells are essentially embedded and nourished by what are just simply referred to as supporting cells. The vestibular, in terms of the mechanism through which the hair cells will depolarize, what we see here in this illustration, we see a single long kinocilium located adjacent to shorter stereocilia. And so the stereocilia must bend in the direction towards this very large, very long, enlarged end kinocilium. When the stereocilia bend in this direction of the kinocilium, that will generate a depolarization. And that depolarization of this axon will then transmit to the cell body in its associated cell body in Scarpa's ganglion that will eventually terminate in the brainstem. So the otolith organs are sensitive to all movements, and that's because of the different orientations. We see the utricle located or oriented in a horizontal type of manner, and we see the sacral oriented in a vertical type of arrangement, making the ability of the otolith organs to sense linear accelerations possible, whether those accelerations are horizontal, vertical, or some combination thereof. When we're talking about these maculae, they do exist as mirror images on both sides of the head. However, in terms of certain types of movements, as we'll see particularly with the semicircular canals, when we have a depolarization on the right side, the left side axons tend to be inhibited and vice versa. Moving on to the semicircular canals, of course, the semicircular canals are paired on the right and left side. Both we see 3 semicircular canals, each on the right and left side, and these canals will sense head rotations in space. And when we talk about head rotations in space, that's because these 3 arched structures are arranged at approximately a 90-degree angle to each other, making them sensitive to spatial motions that are rotation in nature. Just as a review here, all of these axons will project through Scarpa's ganglion to the cell bodies, where another action potential will be regenerated so that that information can be transmitted along the vestibular nerve. We see that within the semicircular canals, the functional unit, the hair cell, exists, but it exists in somewhat of a different type of structure. And the hair cells are contained within cristae, which are found in the ampulla of the semicircular canal. So the ampulla is this enlarged region that houses the sensory organs containing the hair cells within the semicircular canals. The hair cells are contained within a gelatinous medium, as we saw with the otolith organs, except that we don't see the otoconia crystals contained within the semicircular canals. We have the cilia extensions of the hair cells, which give rise to vestibular axons when the cupula has been mechanically disturbed, which occurs with head rotations. And we see, just to finish up here, we have the semicircular canal, or at least a cut portion of the semicircular canal, and the enlarged ampulla containing the cupula. If we take a look at whole body spinning, or just simply head spinning, the central portion here of the functional portion of the semicircular canal is contained within the endolymph, and with head spinning, the endolymph doesn't move, typically the cupula bends in this direction. So here we see a leftward rotation of the head, and cilia bending will lead to action potentials. It could either be, if it generates an action potential, it's activated. If the bending is in the opposite direction, those kinocilia will be inhibited. So a hyperpolarization is induced on the kinocilium. And this depends on the side of the head and the direction of the spin. Typically, when we talk about head spinning, one side of the semicircular canals, on one side of the head is activated and the other side is inhibited. And so in this particular case, we're looking at a left rotating head, and we see the endolymph flow is in the opposite direction. And this illustration will demonstrate the response to the left semicircular canals and the right semicircular canals to a leftward head rotation, whereas the left semicircular canal is essentially detecting or will be producing a movement that is opposite. But just keep in mind that this refers to a leftward head rotation. So as we can see here, if we describe the kinematics of head rotation, we see zero movement, zero speed, and when we spin the head or just result in spinning the body has to go through a period of acceleration, and then we have a constant rate of spinning, a constant velocity. And going through that acceleration period, if we take a look at the firing rate of the vestibular axons from the left-sided semicircular canals, we see this very large deflection. So the impulse frequency starts to increase. And when the body has achieved a set velocity of spinning, we start to see that that firing rate starts to decrease, and it achieves a steady state level which is just slightly higher than the baseline level that was recorded when the head was not spinning. And this is simply a period of accommodation where the neurons cease their fire or slow down their firing rate when the mechanical stimulus has achieved a constant level, and that's velocity in this case. So what we see here, we see that the acceleration, we see another acceleration period in the opposite direction. So basically the person is slowed down to the point where they stop spinning. And because the acceleration is in the opposite direction or detected in the opposite direction, that results in a hyperpolarization of those neurons, and the action potential firing rate drops to close as you can get to zero, where then it will start to fire again to a tonic level. On the right hand side, we see the opposite effect. So because the head is rotating in the left direction, we see the same acceleration period, but we see the firing rate of the right-sided semicircular canal axons drop to close to a level of zero at the start of that acceleration. And that's resulting from the opposite bending of the stereocilia away from the kinocilium, resulting in that hyperpolarization. And we see that during that acceleration period in the opposite direction, this figure is slightly offset here, but we see that there is a period of an increase, a very rapid increase in firing rate of the right-sided axons that will eventually accommodate back to a tonic level. So at this point we'll end part one with the function and the structure of the semicircular canals and the otolith organs, and then we'll address in part two what happens and how we integrate some of these, or how we integrate this sensory information into movement.